Everyone, today we are going to discuss about an interesting topic, that is cell disruptions. Okay, cell disruption. What do you mean by cell disruptions? Means it disrupting the cells for obtaining a intracellular fluids or a proteins. So in the cell disruption process, we are disrupting the cell walls, but we are not disrupting the any other components which is present in the cells. So this method based upon two factors, that is type of the cells, okay, type of the cell and its cell wall. So based upon the cell wall and type of the cells, we are breaking the cells. Okay, moving on to the next slide. So the cell disruptions are two types. Mainly one is mechanical method and second one is the non-mechanical method. First, moving on to the mechanical methods. Mechanical methods, it means we are using some force to disrupt the cell walls without adding any chemicals or any enzymes. So these are the some methods. First one is the mortar and pestle grinding, and second one is the blender, and third one is the bead beating, and fourth one the ultrasonication, and finally homogenization. So next we are discussing about the mortar and pestles. So it is an old grinding method. We are using the plant samples for the cell disruptions. So the plant samples are frozen in the liquid nitrogen. After the cells is disrupted, the metabolites are extracted. Finally, it is dissolved in the organic solvents. That is the main method and the main procedure in the mortar and pestles. Okay, next we are discussing about the blenders. Blenders means what? We are using some high speeds to disrupt the cells for obtaining some intracellular protein. It is almost similar to the centrifugation. Centrifugation means what? We are separating some concentrated materials from the liquid medium. Okay? Next one is the bead beating. That is, we are using some beads like the glass beads or the ceramic beads for cracking the cells and also obtaining the proteins. So we are using the all kind of cells in this method. And the amount of beads, it should be equal with the cell suspension. That is the most important thing in the bead beating. Okay? Next we are discussing next mechanical method, that is ultrasonication. Ultrasonication is also a mechanical method for obtaining the intracellular proteins. It is worked by the vibration. We are using some titanium probe which is immersed in the cell suspension. So here some low shock waves are produced and it leads the cell disruption by changing its pressure. And also it is common in plant cells, fungal cells, because one disadvantage is also there, that is it is very expensive also because it is need a separate room and it is very loud. That is the main disadvantages. Okay, next we are discussing about this is the image of the ultrasonicator. This is the sonicator. This part is ultrasonicator generator. It is a tip. This part is the cell suspensions. Okay. Next we are discussing the homogenization process. It is almost very familiar in our students because in earlier food and microbiology you already studied this part, like where in milk part. Okay. Next, we are commonly use the liquid based homogenization. For what? For the small volumes and cultured cell disruption, we are using the liquid based homogenization. Actually, the cells are lysed by a force, that is a mechanical force. It disrupts the cells through a narrow space. Actually, here it is similar to the bead beating method. So the homogenization is performed by squeezing the cells. Okay, squeezing the cells through what? Through a narrow tube. So it is smaller than the bead beating. So these are the examples, these are the types of the mechanical method which is related to the cell disruption method. So next we are discussing about the non-mechanical method. Okay, the non-mechanical method is classified into three, that is physical method, chemical method and the enzymatic method. In physical method, which contain the freeze-thaw, microwave and osmotic shock and the electric discharge. So first we are discuss about the freeze-thaw. What is freeze-thaw? Okay, in freeze-thaw, commonly used in the soft plant and the algae. It is achieved by the freezing. Okay, it is achieved by the freezing. So it freezing and also it forms some ice crystals and it's expand upon the thawing and ultimately it causes the cell wall rupture. So it leads the cell disruption. Also we can help to obtain our intracellular proteins or fluids. Is microwave, that is in microwave is always similar to the autoclave. We are using some high temperature to disrupt the cells. Also it break the bonds between the cell walls and also it denature the proteins. So it is a risky method because it also disrupts the other components which is present in the cells. Next we are discussing about the osmotic shock. Osmotic shock is always related to osmosis. That means water is entered or moved into the cell and it causes increasing the volume of the particular cell and at one point it bursts out. So it is commonly used in protozoa animal cells. Third is the electrical discharge. So in electrical discharge method is commonly used in the mammalian cells because it achieves the cell disruptions by the electrical discharge. And in the plant cells have no cell walls, but in the case of mammalian cells, it is the cells are bounded by the plasma membrane. So in this method, some of the researchers is allowed to examine the secretion by the exocytosis. And next method is the chemical method. In chemical methods, we are using some organic solvents like surfactants, methanol, benzene, ether, toluene, DMSO, phenyl ethyl alcohol. It is commonly used in the plant cells. But in the case of gram-negative bacteria, we are using the EDTA. EDTA is disrupt the cell walls of the gram-negative bacteria. And the gram-negative bacteria cell walls containing some lipopolysaccharides that stabilize some cations like magnesium, calcium. So this EDTA will chelate the cations and leaving the holes in the cell walls. This is about the chemical method. Next, moving on to the enzymatic method. Enzymatic method, we are using some enzyme to disrupt the cell walls, that is protease, mannose and the glycanase. Glycanase means beta-1,6 and beta-1,3. So it is commonly used for the isolating the cells without the protoplast. So these are the cell disruption method. So cell disruption method is mainly two types, that is mechanical and non-mechanical. And mechanical means, and again non-mechanical is classified into three types: physical, chemical and the enzymatic. And the physical method which containing the freezing and freeze thawing and the microwave, etc. Okay, hope you guys are understood. That's all.